Hi, I'm Vladek Sandler, co-founder and CEO of Hemogenyx Pharmaceuticals, and we're here for our lab meeting to discuss the progress of our CBR program and some other programs. And I'm here with pretty much everyone in the company. Left to me is our director of preclinical development, Elena Shrestha, and our principal scientist, Ronan Ben-Yehuda, and other people. And these people will actually be featured in a series of videos that will describe the work of our company and our different projects and the role of these people in the development of these projects. Thank you so much.
Hi, my name is Ronan Ben-Yehuda. I'm a principal scientist at Hemogenyx Pharmaceuticals. I joined the company about 4.5 years ago. Prior to this, I was working on my PhD in the Technion in Israel, where I was studying the electrophysiological and metabolic abnormalities of induced pluripotent stem cell-derived cardiomyocytes that were generated from patients carrying a PRKAG2 mutation. Additionally, I was able to show that using CRISPR-Cas9, after correcting the mutation, all the abnormalities were completely eliminated. Here at Hemogenyx, we focus on the development of immunotherapies for blood diseases, including acute myeloid leukemia. As a principal scientist, I oversee all of our research endeavors, and I'm happy to share that we recently submitted an IND application to the FDA for our CAR T therapy for acute myeloid leukemia. To do this, we have developed an entire process in-house for cell manufacturing in our clean rooms. We did process development, process qualification, and an establishment of a complete quality system, including all SOPs and master batch records. Additionally, we have established a GLP facility for testing before the drug product could be released. Additionally, I want to share that we are working on another exciting project, our CBR, that stands for chimeric beta receptor. In this project, we're utilizing the innate part of the immune system in order to target cancer cells and infectious diseases such as viruses, including SARS-CoV-2, Zika, Ebola, and other virus types. We are about to start a BSL-3 facility experiments in order to test our constructs in vivo in mice. For that, we will be using a live replicating virus. And as always, we will share our results and news with you. Thank you.
Hi, I'm Michael Hogbro. I'm a senior scientist here at Hemogenyx. I joined in June of 2021, and before that I was getting my PhD in chemistry from Princeton University with a focus on protein chemistry. I was working on novel methods for in situ protein semisynthesis, and decided after I graduated to join industry, and at the time I found Hemogenyx, and I thought it was a very exciting opportunity and really a company that was uniquely situated to have a large impact in the immuno-oncology field. And I was excited to join and bring some of my expertise in protein engineering, and I've been able to do that here, and I've been working on some of our biologics, especially some of our antibody drug conjugates. I've also been taking part in our CAR T therapy, which is our most advanced candidate. We just submitted an IND application to the FDA for phase 1 clinical trials, and we are doing the manufacturing for that cell therapy here in our facility in Manhattan. And I was a part of the team that optimized the manufacturing process and performed a number of the process qualification runs. And so it's really exciting as we wait to hear back from the FDA on when we'll be able to begin actually treating patients and seeing some of our hard work in the lab really translate to helping patients. Thank you.
My name is Elena Shrestha. I'm the Director of Preclinical Development at Hemogenyx Pharmaceuticals. I've been with the company for 5.5 years and have had the opportunity to witness and be part of the incredible journey the company has taken over the years, starting with 2 people to now over 12 people. Before coming to Hemogenyx, I received my PhD from NYU School of Medicine in Cell and Molecular Biology. There, my research focused on understanding the transcription regulation in macrophages by liver-X receptors and its implications in lipid metabolism and diabetic atherosclerosis. At the time, I was hugely fascinated by all the developments in immunology, immuno-oncology, and stem cell biology, and that's precisely what attracted me to Hemogenyx, as its core concentration is in immunology. I joined Hemogenyx as a scientist, but over the years my role has evolved, and now my role is focused in overseeing all functions of CAR T development as well as bispecific development, the bispecific antibody development. And both of these therapies are developed as a treatment for relapsed refractory AML. A big part of my role is to also facilitate the transition of lead drug candidates into clinic by identifying the most effective roadmap for drug development, while also ensuring that the quality of the drug product is our top priority. With regards to the bispecific program, I was heavily involved in the preclinical characterization of our CDX antibodies in both in vitro as well as in vivo models. The time right now is very exciting for our bispecific antibodies, as we've already begun the development activities, starting with the cell line generation. The purpose of this is to develop a master cell bank which will eventually be used for downstream antibody production. And this, together with completion of safety tox studies, will allow us to prepare an IND, which will allow us then to enter phase 1 clinical trials with our bispecific antibody to treat relapsed refractory AML. On the CAR T front, this year has been a remarkable year as we transition from research into development in preparation for our investigational new drug application with the FDA. We also moved last year from Brooklyn to a brand new state-of-the-art facility in New York City, which also consists of 2 clean rooms. One of the most remarkable achievements we've had this past year is that we were able to successfully set up our own in-house GMP facility, which is now fully functional and qualified for use in manufacture of our CAR T cell therapy product. We were also able to develop our process for large-scale autologous CAR T manufacturing, as well as develop part of the analytical testing in-house. And obviously, all of this takes an incredible team effort. It's an enormous team effort. We have an incredible team, so kudos to our incredible team. This wouldn't be possible without them. So I honestly think our team dynamics is incredible, and this is a true reflection of Hemogenyx's core values in team building and team evolution. And I'm forever excited to see what the future holds for the company, and we need to just keep making strides in developing new novel therapies to treat deadly diseases that are out there. Thank you so much.
Hi, my name is Rocio Moron. I'm a lab manager and company coordinator at Hemogenyx Pharmaceuticals. I joined in September of 2022 after I graduated from St. John's University with a bachelor's in biology. As a lab manager, I support the infrastructure and operations of the lab, as well as I'm involved in the quality department, doing the raw material specifications, equipment files, and any other necessary projects in order to have a fully qualified system, which is what we currently have as a GMP facility. I truly enjoy working in Hemogenyx, the motivation, the team, the managers, the direction that this company is going to. I'm very excited to see what it holds for us. Thank you.
Hi, my name is Satish Dhingra. I joined Hemogenyx exactly 1 year ago as a senior scientist. Previous to joining Hemogenyx, my position was of principal investigator at Columbia University Medical Center, supported by the NIH. I had extensive experience working with drug resistance in malaria. I joined Hemogenyx for 2 reasons. The first reason was it gave me an opportunity that I could work in immuno-oncology field, which really directly impacts patients' life here in America. The second reason was the opportunity to work in two very unique projects, one of them being the CBR project. The aim of the CBR project is to develop a uniform or a unique therapy which can be utilized to target multiple infectious diseases using the same therapy platform. So I've been working on that for the past 1 year. Secondly, I've been also heavily involved in our CAR-T project, for which we are about to start phase 1 clinical trials soon. My responsibilities for that project were to develop analytical methods that will be used to release patient samples or patient therapy product, and we've developed them in-house. Thank you.
Hi, I'm Stuart Tench, Director of Quality at Hemogenyx Pharmaceuticals, LLC. I joined in August 2022 to create the quality management system here at Hemogenyx. Since I joined, we've qualified our clean room to manufacture our CAR-T in phase 1 and wrote SOPs in order to manage the quality system, as well as our CAPA system and other quality risk management systems. We have qualified our analytical test methods in order to release our CAR-T product, and we are looking forward to having an IND approval and manufacturing our CAR-Ts.
Hi, I'm Anu. I am a scientist working at Hemogenyx. A little bit about me: I went to the Georgia Institute of Technology to do my undergrad. I got my undergrad in chemical and biomolecular engineering with a concentration in biotechnology. So naturally, when interviewing for the company, I was kind of interested on the pharma side of things and getting into the pharmaceutical field. And when I interviewed for the company, I actually joined the company as a lab manager 2 years ago in May 2021. And after working for the company for about a couple of months, and because we're such a small company, I was helping out a lot in terms of the experiments we were doing and a lot of the projects as well. And in that path, I found out that I was really interested in getting involved in the science of everything. In terms of my background, because I did chemical engineering, I didn't take too many science classes or biology classes, so it was a learning curve, but I was ready to take on that. And then I switched to becoming the scientist in May of 2022, so last year. And since I started working as a scientist, I've worked on various projects, including a bispecific antibody project. That's where I started. And then working on that for a couple of months, and then I switched to the CBR project and was working alongside Satish to learn how to package the plasmids and create lentiviruses. And after that is when we started getting the whole company involved in our IND for our product and our clinical therapy. And so I was working on the analytical team, so doing analytical testing for all of the drug products that were being made from our different PQ runs. So I was working on that and doing all the documentation, and now that the IND has been filed, I am back in the lab continuing the research alongside Satish and Ronan and everyone else on the CBR project. And that's what I'm doing right now. Thank you.
Hi everyone, my name is Louise Pham. I graduated with a master's degree in biomedical engineering from Cornell University in 2020, and right after that I had an opportunity to join Hemogenyx Pharmaceuticals as a research associate, and since then I have been a part of the company, and I'm really happy to be able to contribute my effort and knowledge into creating cell therapy and a drug candidate to save lives. And my everyday at Hemogenyx is very exciting. We will come in and discuss multiple projects with my co-workers, and we are very passionate about doing research and come up with different ideas to contribute to the success of Hemogenyx. And I'm involved in multiple projects such as CBR, chimeric B receptor bispecific antibody, and most importantly, our CAR-T project that has been submitted to the IND recently, which is a very exciting news. And right now, with the IND being submitted, we are all focusing on doing other research projects, and I'm very excited to be a part of Hemogenyx. And thanks for letting me introducing myself to everyone.
Hi, my name is Juliana. I'm a cell and gene therapy engineer here at Hemogenyx. I'm a graduate of Stony Brook University. Previous to this, I was working as a cell therapy specialist on several different kinds of cell therapies, CAR-T therapies, CAR-NK therapies, for different diseases. And I came to Hemogenyx. I was really excited to join. There's so many opportunities here to learn and also to really work on amazing novel cell therapies. Recently, I helped set up and validated clean rooms for our GMP environment, as well as an environmental monitoring program, which I created and implemented. And we recently submitted an IND for a CAR-T therapy that will help treat acute myeloid leukemia. More recently, I've been more involved with research now on the CBR project that we have going on, and I'm excited to be working for a company that can help a lot of people and will hopefully be very, very positively impactful. Thanks for taking the time to get to know me.
Hello, my name is Deirdre. I'm from Ireland, which is where I studied and graduated with a degree in life sciences. I then began my career in the pharmaceutical industry in Ireland. And this is when this fueled my passion for drug development and pharmaceutical development, which is why I recently made the move to the United States and joined the team at Hemogenyx. At Hemogenyx, my main role here is predominantly to learn the manufacturing process for our new CAR-T product, which we have just submitted the IND for, so it'll hopefully be going to clinical trials very soon. In the meantime, I'm working as well with some of the scientists to bring novel therapies forward to treat different diseases. So I'm really excited to see what the future holds for myself and everyone at Hemogenyx, and to see what we can bring forward to help and improve patients' lives in the future. Thank you.
Hi, my name is Jessica Jang. I am a senior scientist at Hemogenyx. I received my PhD from University of California, Riverside, where I investigated the innate immune response to a number of infectious diseases, from helminth infections to toxoplasma to even intestinal bacterial infections. After I graduated with my PhD, I also worked at Celgene, where I was in the translational development department. There I studied the mechanisms of action and mechanism of resistance for small molecules to target acute myeloid leukemia and myelodysplastic syndrome. After Celgene, I worked at Selectus, where I used gene editing mechanisms in hematopoietic stem cells to correct monogenic diseases. This work focused on the advantages of using hematopoietic stem cells, which is the ability to differentiate into any number of immune cells. At Hemogenyx, my current project focuses on differentiation of macrophages from either hematopoietic stem cells or blood monocytes, so we can re-engineer these macrophages to express the chimeric B receptor and treat infectious diseases. This platform hopefully will allow us to expand to other diseases such as cancer or solid tumors. I'm really excited about this project because I think it has the potential to be used in many different diseases, not just infectious diseases. Thank you, and I hope you learned a lot about Hemogenyx today.